Hi, I'm Xinhe Zheng, and I'm an undergraduate at UC Berkeley. The paper I'll be presenting today is titled as Differentially Expressed Markers Between Chemoresistant and Non-Chemoresistant Cells in TNBC. First, introduction. So this paper focuses on chemoresistance in TNBC, and it employed TNBC single-cell RNA sequence from dataset GSE118389, and I've discovered 1,987 differentially expressed markers between a chemoresistant cluster and non-chemoresistant clusters. Some background. TNBC are cells that lack receptors for estrogen, progesterone, and HER2 protein. Is relatively uncommon, as only 15% of breast cancers are TNBC. Chemoresistance. Chemoresistance happens when cancers stop responding to chemotherapy. It's also a possible cause of the metastatic relapse. From the literature review, most studies focus on therapeutics or the mechanisms of chemoresistance of one or a few genes, whereas only a few study focuses on a systematic list of chemoresistant markers. The methods, first the data. There are 6 primary non-metastatic and triple-negative breast cancer tumors from females in the study, and their epithelial cells are divided into 5 epithelial clusters with a density-based approach that automatically selects the number of clusters. It's performed with monocle in R. And of the 5 clusters, cluster 2 expresses various signatures of chemoresistance. Then data processing and differential gene expression analysis. So first, epithelial subpopulation is subsetted, and it's then separated into basal epithelial subpopulation and luminal epithelial subpopulation. Also, cluster 2 is isolated with the threshold of average expression greater than or equal to 1 in at least 3 markers from the top 10 markers that characterize cluster 2. Then the differential gene expression analysis is performed between cluster 2 and other clusters in all of the 3 subpopulations. Then the results. So here are the differentially expressed markers between chemoresistant, which is cluster 2, and non-chemoresistant clusters in epithelial subpopulation. There are 17 markers with a p-value less than 10 to the 40th power. For example, EEF2, RPL3, and CSRP1. And here are the differentially expressed markers between chemoresistant and cluster 2 and non-chemoresistant clusters in luminal epithelial subpopulation. There are also 17 markers, but with a p-value less than 10 to the -5th power. And for example, we have CDC74B, DHRS2, and THRSP. Now discussion. So these are the functions of some of the significantly differentially expressed markers. First, EEF2, it mediates the translocation phase of protein translation. Also, in higher eukaryotes, EEF2 may be one of the important elements in regulating protein synthesis when there is a lack of energy. Then RPL3, it's an essential regulator of cell cycle apoptosis and DNA repair. It also regulates the expression of P21 in response to chemotherapy. It can also control DNA repair independent from the cell's P21 state. CSRP1 mediates protein-protein interactions and is crucial for cell differentiation, cytoskeletal remodeling, and transcriptional regulation in vertebrates. It's also a tumor repressor for colorectal cancer and could be a diagnostic marker for liver cancer. Some limitations in the study, the sample size is small. It only contains 695 epithelial cells from 6 TNBC tumors. Also, the threshold for the presence of markers were raised for subsetting the epithelial subpopulation and cluster 2 cells to lower the false positive rate. So some cells may have been subsetted into other clusters or other subpopulations, which may cause inaccuracy. Finally, conclusion. So 1,987 differentially expressed markers are found between a chemoresistant cluster and non-chemoresistant clusters in epithelial subpopulation. Also, 17 were found in luminal epithelial subpopulation, and there is a considerable overlap between these markers. And thank you for listening.